Good afternoon, everyone. I'm absolutely delighted to see you all here this afternoon. My name is Richard Pickersgill. For those who don't know me, I was the former head of SBBS and currently the Deputy VP Science and Engineering. Inaugural lectures are always a great delight. It's an opportunity for a colleague to give insights into the work they're doing in an accessible format. It's an opportunity for us all to celebrate their achievements and successes. This afternoon we've got a really wonderful lecture from Steffi, and we shall talk a little bit about that in a moment. I should give a little bit of housekeeping, two things. That we're not expecting any fire alarm, and if you hear a fire alarm we will escape through the doors and we'll get to Library Square. And secondly, I think if you put your phones on silent, that would be nice during the lecture. So we're gathered this afternoon for the inaugural lecture of Professor Steffi Krause, who's just putting a mobile on silent as we speak. Steffi joined the School of Engineering Material Science in 2004 as an EPSRC Advanced Fellow, and I have had the pleasure of knowing Steffi from the time when she was collaborating with Mike Watkinson, the chemist using click chemistry to pattern self-assembled monolayers, and Mike is with us today. Hello, Mike. Engineering is one of the flagship research areas in science and engineering at Queen Mary. And Steffi's journal papers contributed to our submission to the most recent research assessment exercise, REF 2021, having the second highest ranked papers in the UK. And to the overall ranking of engineering at Queen Mary as 7th out of 89 submissions. 7th in GPA, which is the key metric. In terms of collegiality, Steffi has been Director of Academic Standards in SEMS for many years, a role which she has helped steer the examination processes from being predominantly in person, through entirely online during the COVID period, to the current authentic assessment approach. Through this, she helped SEMS develop the online examination system that controls all modules through the assessment cycle. And Steffi currently lectures clinical sensors and measurements, which seems to be entirely appropriate. At Queen Mary, Steffi has received over £2.4 million of funding from EPSRC, BBSRC, and EU for her work on photoelectrochemical imaging, of which we will hear more during her lecture. It's always interesting to see the arc of our colleagues' work presented in these inaugural lectures, and today Steffi will take you on a journey from the development of sensors for single analytes to the imaging of electrical and electrochemical cues from living cells. Steffi was promoted to Professor in Electroanalytical Systems in 2019, so arguably she's had plenty of time to prepare this inaugural lecture. The lecture this evening will be around 45 minutes, and if there's time at the end, we're running a little late, Steffi is happy to take questions. And if there are further questions, of course, we will have a reception in the foyer outside, so you can take the opportunity then to ask questions. We have special visitors here today, so I'd like to welcome family and friends here to Queen Mary. I extend a very warm welcome to them. A very warm welcome. Steffi has asked Professor Karen Hing to say a few words. Karen Hing is Professor of Biomaterials and Tissue Regeneration, and Steffi and Karen have been collaborating on the development of electrochemical cell imaging technologies for, I'm told, the past 10 years. So let me conclude my welcome address on behalf of the university and hand over to Karen, and I will be back for the question and answer session.
Okay, thank you, Richard. And it's my honour to be able to introduce Professor Steffi Krause tonight. Steffi hasn't only worked on sensors that see, but she's also worked on sensors that smell and taste as well. Her exploration of the sensors field began with her PhD in 1991, back in the days when we were running around in fields in day-glow outfits. Steffi, though, was at the Humboldt University of Berlin working on the development and characterization of sniffing sensors, so field effect oxygen sensors under the supervision of Professor Werner Moritz. She successfully completed her PhD in 1994, and having lived at home during her undergraduate and postgraduate studies, was now clearly keen for a change of scene, and won a prestigious DARD scholarship, a scheme that enables young German researchers to gain academic experience abroad. She seized this opportunity to work in a new and exciting location, travelling to the balmy climes of Newcastle, where she joined the group of Callum McNeill at Newcastle University as a DARD Fellow from 1994 to 1996. Also moving on to working on sensors that now taste, developing biosensors for urea and creatinine detection based on impedance changes caused by the degradation of thin films. So Steffi clearly enjoyed being part of the British science community. Some of that might have had something to do with a certain on. Choosing to accept a postdoctoral research position in John Cooper's group at the Glasgow University from 1996 to 1997. Here, she expanded her knowledge in the biosensors field and focused on the development of nanostructured electrodes for measuring the fast reaction kinetics of single-cell bioassays, before gaining a lectureship position at the chemistry department of Sheffield University, where she continued her work on biosensors and started her work on imaging with light addressable potential metric sensors. In 2002, she was also awarded a prestigious EPSRC Advanced Research Fellowship and continued to work on these photoelectrochemical imaging techniques up at Sheffield for a further 2 years, before wisely using her fellowship to leverage herself a senior lectureship position in 2004 down here in London at Queen Mary, within the Department of Materials as it was back then. Since then, Steffi has become a valuable member of the senior academic team within SEMS. Known for her fair and forthright temperament and a commitment to EDI, she led SEMS' first Athena Swan application, which was awarded in 2013. And as already mentioned by Richard, as Director of Academic Standards, has worked to implement and simplify our robust procedures to ensure that we at SEMS maintain high levels of standard in academic integrity and delivery. However, getting back to the science, since arriving here at Queen Mary, Steffi has established herself and her team as internationally leading pioneers in the field of photoelectrochemical imaging, including driving the development of the technology to facilitate real-time imaging of live cells. And believe me, doing anything real-time with live cells is a huge challenge. So she's gone on to publish 24 internationally leading papers in photoelectrochemical imaging, while also maintaining interests in international recognition in the development of both chemical and biological sensors. And this is reflected in her role as the conference organiser and the host of the 16th International Workshop on Engineering of Functional Interfaces, which is taking place this summer here on campus at Queen Mary in July. So, with great delight, I'd like to introduce Professor Steffi Krause.
Thank you very much for this really nice introduction, Richard and Karin. And thank you very much for coming today. It's a real delight to see so many familiar faces, and also in person and also in line. And what I want to do today is I want to take you on a very personal journey, starting from the sensors for single analytes that I started to develop in my PhD, going to use one of these sensor principles and developing it into an electrochemical imaging technique that allows us to see things like, for example, living cells. So just to start at the beginning, I grew up in Berlin. So, and while I was going to school in Berlin, I had an early interest in chemistry. So there was a lot of cool chemistry around in the 1980s. Just to give you a few examples, there was a first breakthrough in high temperature superconductors in 1986, which basically eventually led to modern MRI machines and maglev trains. There were the Buckminster fullerenes were for the first time described in 1985 in an English-speaking journal, I should add, and that sparked a lot of nanotechnology and materials like graphene later on. Also, the polymerase chain reaction was for the first time used for the amplification of DNA, and of course that is the foundation of all the modern advances in forensics, medicine and genetics that you continuously see on television and in the news. And finally, there was also the first modern lithium battery described with a carbonaceous anode rather than a lithium anode, which you nowadays find in your phones, in your laptops, and also in electric cars. So therefore, because of all that chemistry around, and since I was interested in chemistry, I decided to study chemistry at the Humboldt University in Berlin. In the top left, you can see the historic chemistry building where we did most of our experiments in the first and second year, and also listened to lectures. But unfortunately, by the end of the first year, I had realized that maybe I'm not cut out to be a hardcore synthetic chemist, because you were actually charged for the glassware that you dropped and smashed, and I had a rather large bill at the end of the first year. And also, every time I took a laptop, one of my lab coats out of the washing machine, it looked like that. So I decided that synthetic chemistry is not for me, and I might have to dedicate myself to a more physical branch of chemistry. So I spent more time in the Walter-Nernst Institute for Physical and Theoretical Chemistry, which is in the Bunsenstraße in Berlin. And essentially what I did right from the start of the second year undergraduate studies, I started to help out in the group of Werner Moritz at the Humboldt University in the Institute for Physical and Theoretical Chemistry, and this group was working on chemical sensors. So bear with me while I explain to you how these sensors actually work. So these were einselective field effect transistors. So you know probably that field effect transistors are on all your electronic devices, and essentially you have a semiconductor substrate, like for example p-type substrate. You have two n-doped areas, which are the source and drain, and essentially on the top you have a gate insulator and a gate electrode. So the voltage applied to the gate electrode controls the current that flows between source and drain. So above a certain threshold voltage, you can see that basically there's an N-type channel at the surface of the semiconductor, and that allows a current to flow. Now, in 1970, so I'm going back even further, a scientist called Pete Bergfeld invented the first ion-selective field effect transistor. So he used a transistor without a gate electrode and used the electrolyte as the gate electrode and applied the gate voltage through a reference electrode that was in the solution. So if you have, for example, a change in pH in the solution, so let's say you make it more acidic, then protons will absorb onto the surface of the gate insulator, which is typically an oxide, and that will cause another charge on the surface, which causes a shift of the current voltage curve along the voltage axis. So essentially this is a potentiometric sensor that where you can measure, that you can make basically sensitive for all sorts of different ions. So in the group of Werner Moritz, we basically coated the gate insulator with material called lanthanum fluoride. Lanthanum fluoride is a fluoride ion conductor, and that made these types of sensors sensitive for fluoride ions. So I thought when I joined this group and helped out in my free afternoons in the second year as an undergraduate, I thought this is super cool. Lots of different disciplines were thrown together. There was some electronics, there was some material science, there was some electrochemistry. So I thought this is a really cool area of research. It also allowed me to make an unexpected link to my dad, who had a degree in electrical engineering. And for the first time, he had the impression I was actually taking an interest in this area of expertise, because I got him to explain to me how a field effect transistor works. Very important in a day and age where you can't Google, where you couldn't Google things like that. So what are these ISFETs good for? So as I said, they can be made sensitive for all sorts of different ions. So they're very useful for biomedical and healthcare applications. So for example, you can use them for measuring blood electrolytes, for blood pH. You can use them for environmental monitoring, so water quality, drinking water, and also water quality or the wastewater quality, so measuring things like pH, fluoride, and heavy metal ions. You can also use them partially for gas sensing, because some gases, when they dissolve, they form ions, like for example HCl, HF, and also ammonia and carbon dioxide. And then there are so lots of industrial applications where you can use them in fermentation processes, for example. So lots of useful applications. So I really enjoyed the work in that group, so I decided to stay for my PhD. So in my PhD, I actually worked on to develop a field effect oxygen sensor. So there is not this is not a field effect transistor, but it's still called the field effect structure. It's much simpler. It's a capacitive structure. So this time we stuck with the lanthanum fluoride, but we completed the structure with a platinum gate electrode. I'm not going to bore you with exactly how we did the measurements. But maybe a note on why would we even bother with gas sensors? Well, there's a lot of health and safety issues that can be controlled via gas sensing. So, for example, in the UK, until the 1980s, people used canaries and white mice as a warning of poisonous or asphyxiating gases. So, and only in 1986 were they actually phased out and replaced with the electronic sensors. So making gas sensors is a very worthwhile undertaking. And why oxygen? Well, oxygen is also, of course, if there is not enough oxygen there, then this has an asphyxiant effect, or if you have too high an oxygen concentration, there's a fire risk. And then there are lots of different applications. So, for example, in fuel cells, you have to control the oxygen concentration. For things like anesthesia and ventilators in hospitals, you need oxygen sensors. Of course, you need to also control the atmosphere in space stations and in aeroplanes. And of course, food packaging is actually quite an important application, because a lot of times, or many times, the food is packaged under inert atmosphere, so to stop it from oxidizing, to make it last longer. And of course, there's again the fermentation processes and cell culture, where you need to control the oxygen. So lots of different applications. And my research on this was quite successful. So you can see me there in the top right-hand corner in front of my gas sensor setup with a state-of-the-art PC. And basically it was very successful. We managed to detect over 8 orders of magnitude of oxygen with the sensor. Then I managed to I got to go to Moscow to the Kurchatov Institute, and there I managed to do a few experiments where we actually found out that the sensor also responds to fluorine and to hydrogen fluoride. Very dangerous experiments. And then when I came back, we also tested it for hydrogen, and it also responded to hydrogen. So I was starting to wonder, if I spend a few more years, maybe it will respond to every gas there is. But I I should not be so disparaging about that, because later my PhD supervisor developed this into a very effective sensor as an early warning sensor for forest fires. So I should not disrespect this type of sensor. But I decided it would be really nice to move on into an area where maybe the sensors are a little bit more selective. And while I was going to conferences during my PhD, I saw a lot about biosensor development. So biosensors usually use biomolecules like enzymes, antibodies, or DNA, and that makes them very, very specific to the analytes that you want to detect. And usually these are coupled with a transducer, which could, for example, be an electrode. So the most well-known biosensor, or the most commercially successful biosensor, is of course the glucose biosensor, because there is such a high incidence of diabetes. Diabetes increased from 200 million in 1990 to 830 million in 22, so that is quite extreme, because people live longer and they're overweight, so more incidence of diabetes. And a lot of these people have to check their blood glucose levels quite regularly. So the disposable sensor market for glucose sensors is huge. So the way they work, just maybe very briefly, because I've got it on the slide, is use the enzyme glucose oxidase, which reacts with glucose and the co-substrate oxygen to produce hydrogen peroxide. And the hydrogen peroxide can then be oxidized at an electrode, and the current that's flowing through the electrode is proportional to the glucose concentration. So very simple electrochemical sensor. So in the 1980s, these types of sensors were for the first time developed, and in 1987 the first home testing kit was launched by a British company, it was called the Medicines Exactech strip. It basically needed about 20 microliters of blood, which was dropped onto these electrodes, which were screen printed, and the result was then announced, you would get the result within 30 seconds. Nowadays, you need maybe only 0.3 microliters of blood for one of these sensors, and you get the results almost instantaneously within 2 seconds. So these were commercially very successful, but there are now lots of commercial biosensors out there. So an example is this product from Abbott, which is the Abbott iStat, which can detect multiple analytes at the point of care. So you basically buy these little cartridges, which are for different groups of analytes, like blood gases, metabolites, or cardiac markers, or coagulation factors. And you basically put a few drops of blood on there, put it into this iStat, and then the results are transmitted to your PC or to a smartphone. So that is used by doctors, but the glucose monitoring has also made a lot of strides in the last few years. So there is now the possibility of continuous glucose monitoring. So you basically measure the glucose in a sensor that can be put into the arm and stays there for about 14 days. And it can continuously measure the glucose in the interstitial fluid. So that is basically, you only have a short step of pain in the beginning, and then it continues to measure your glucose. So biosensors have made huge strides, and even in the 1980s and 90s, the research in this area was very exciting. So I decided I wanted to work on biosensors. So I decided to apply for this DAD stipend and move to the University of Newcastle to Callum McNeil's group. And in that group, I learned a lot about biosensors. I learned a lot about clinical biochemistry while I was there, and I also learned how to have a good time, actually. As you can tell from the picture. And I then moved on to John Cooper's group in Glasgow, as Karen has already mentioned, and I spent a year in the clean room fabricating nanostructured electrodes for single-cell biosensors. So that was the first time I actually worked with living cells, although mostly spent time killing them, but it was still an experience. I then moved to my first academic position in Sheffield, and then I was thinking, of course, how can I, I continued my work on biosensors, but I was thinking, how can I actually combine all the different experiences I had? And I was thinking that it would be really cool if I could do measurements with living cells. So this is what I set out to do. And during my PhD, I had already come across this technique, which was first published by Hafmann. This is basically called light addressable potentiometric sensors. So now you will realize why I was talking so much about field effect structures earlier, because this is again a field effect type of sensor. So we have essentially a silicon substrate with an insulator and maybe a sensor material on top. And again, we have the possibility to apply a voltage through a reference electrode, and that produces a depletion layer inside the semiconductor. So at the bottom left, you can see the band model of this type of device. So if you shine light onto the structure, what happens is electrons are promoted from the valence band to the conduction band, and the minority charge carriers will accumulate at the interface to the insulator. And if you turn the light off, what happens is that these charge carriers go the opposite direction and they will eventually recombine. So what that means is if you modulate the intensity of the light, so continuously turn it on and off, you basically get an AC current that's flowing through the structure. And the special thing about it is that the current is only flowing in the area that you have illuminated with the light. And that gives a lateral resolution. So you can basically address every spot on the sample with light. Very similar to the ion selective field effect transistor, these types of sensors are very sensitive to charge. So they can be used for measurement of local pH and ions, and also for local electrical potential measurements. But we had the idea to also use this for impedance measurements. So if you look at the characteristic curves that you measure, so current voltage curves again, you can see you get the sigmoidal shape. You can, if you measure ion concentrations, you get again a shift of the curve along the voltage axis. So this is a potential measurement. But you can also look at the changes in the maximum current, which basically tells you about the local impedance, so local resistance and capacitance of any material that you have on top of your sensor. So I wanted to work with living cells, so why electrochemical imaging? Because if you look around, you will see lots of really fancy fluorescent picture of living cells, and you can do all sorts of sensing with fluorescent markers. And the question is, if you can get beautiful images like that with lots of information that it gives you, why bother with electrochemical imaging? Well, there are some limitations to the fluorescent microscopy. So fluorescent dyes, for example, suffer from photobleaching, so the fluorescence will decrease with time. Fluorescent dyes can also interfere with the biology that you want to detect. And of course, if you want to make cells fluorescent, you have to genetically modify them. So that makes things like long-term monitoring difficult. And of course, if it comes to functional imaging, like measuring things like electrical potentials, concentrations, and also charges, then electrochemical methods are far superior to any optical techniques. And that's why we wanted to use these light addressable potentiometric sensors to look at cells. So we also looked at existing technology, of course, what's out there, what are people using for electrochemical measurements with cells. And these are typically multi-electrode arrays. So you can see I've taken a picture out of a paper from 21, so they're still in use. And these electrode arrays, you can see there are individual little circular areas. This is where the exposed electrodes are, and there's a certain spacing between them. So if you enlarge that, I don't have any pictures of cells on there, but if you imagine you put a cell, you start growing cells on them, some of the cells might end up directly on these electrodes, but then some of the cells may only partially cover them. So how do you interpret the signal then? And it becomes more difficult. And then, of course, many cells will just grow in the gaps between the electrodes. So not ideal. With our light addressable potentiometric sensors, we can actually address a specific point on the sample, so we can seek out the cells, we can image them, and that is the advantage why we wanted to use these light addressable potentiometric sensors. Also, by the way, this fabrication of these multi-electrode arrays, because it's multi-step fabrication, is much more expensive than having a simple semiconductor substrate with an insulator on top of it. Okay, so what did we do? We wanted to, some of the problems that we had to solve were the resolution of the light addressable potentiometric sensor, because when I first came across this technique, the resolution of these types of measurements, if you wanted to see a single cell, was totally impossible. The resolution was hundreds of micrometers, and we wanted to improve that resolution. So we found, we basically recognized quite early that we couldn't use the thick silicon substrates that everybody else was using, because you get a degradation in the resolution because charge carriers are diffusing laterally in these thick silicon substrates. So we started using very thin layers of silicon. So these are basically 1 micrometer or half a micrometer thick layers of silicon on a transparent substrate, sapphire. And we then also, what we also did is we replaced the traditional insulator with a self-assembled organic monolayer. So this is essentially, if you wash the surface of silicon with HF, you get a hydrogen-terminated surface that can react with a double bond and then give you a nice self-assembled organic monolayer. And the advantage is, first of all, you have low density of interface states, which makes for very good photocurrent measurements. They're very thin, of course, you get large photocurrents, and they're also terminated with functional groups, so you can bind all sorts of things onto the surface that you can then use for different sensing applications. We've also played around with the wavelength that we used. So traditionally, you would just use light that has an energy greater than the band gap of your semiconductor, so to promote electrons from the valence band to the conduction band. That's so-called single photon excitation. But we also thought maybe we can improve the resolution by using 2-photon excitation. So in this case, you use light that has an energy smaller than the band gap, which means you have to absorb 2 photons simultaneously. So the advantage is that if you focus, for example, a laser onto the substrate, you will only get a photocurrent excited in the focus where you have a high enough concentration of photons. So basically, with using a 2-photon excitation, we improve the effective laser beam profile for charge carrier generation, but maybe more importantly, the light reflected and scattered in the structure will not cause any photocurrent. So just to show you, so when we used the wavelength of 1250 nanometers with our silicon substrates, we got a pure 2-photon effect, as you can see from the intensity dependence of the photocurrent. So now, how did we actually do the measurements? So this is basically our first, one of our first setups. We still have one of these setups that we have in regular use. So this is an example where we used a diode laser. So we cleaned up the laser beam with a spatial filter and then focused it through the sapphire into the silicon. And the measurements now are done with a lock-in amplifier. And the way we did it is you basically move to a point, you measure, move to the next point, you measure. So it's a very, very slow process because you have to basically very precise, you get very precise photocurrents in each spot, but you move the stage, you stop, you move the stage, you stop. So very relatively slow imaging, but at least we managed to achieve the resolution that we were craving. So this is the example where we basically used a photoresist pattern as our model. So we wanted to check out the resolution with that. So we imaged this photoresist pattern, as you can see here. That's one of our photoresist patterns. And when we then scan the laser beam across the edge of one of these photoresist patterns, we can estimate the resolution. And with the 2-photon excitation, we indeed got a very high resolution of less than a micrometer. And with a single photon excitation, the best resolution we got was about 1.5 micrometers. So we had the resolution, even though this temporal resolution was relatively poor. So it took, with this setup that I showed you in the previous slide, it takes maybe 10 to 15 minutes to record an image like that. So if you want to work with living cells, really you want to be a bit faster, because things might already be happening, the cells might have already died and gone away by the time you finished your image. So we really wanted to measure faster. So we were scouring the literature a bit to see whether we can, what's out there. And I saw this paper by Anirban Das, where he basically used an analog micromirror to move the laser beam across the sample. So rather than moving the stage step by step, where you move the electrochemical cell, we actually move the laser beam with the micromirror. But unfortunately, the resolution was really poor. What was really fortunate is that Anirban contacted me, and we applied together for Marie Curie Fellowship. So he came to London and he built us this setup, which basically combined his mirror scanning with a very high resolution. So essentially we have the scanning mirror here. You can see this enlarged in that little animation on the top. So when you move the mirror, you basically also move the focus across the sample. And this is a very precise 4F design that you also find in laser microscopes. You have a scan lens, a tube lens, and a microscope objective, which means you can scan the focus without degrading the resolution and without losing intensity. So that's very important if you want to do imaging. And to show that he can do it quickly, he basically injected an HCl solution into a pH 7 buffer. So these are images that were taken 8 frames per second. So this is in real time. If you look at that little video, you can see that the pH is going down as the HCl is added, and then the buffer solution neutralizes the acid again. So it's a very fast measurement, and we were quite happy with that. So we explored various sensor applications. So this on the right-hand side, you can see the work by Jian Wan, and she basically tried to measure DNA. What she did is she microcontact printed a positively charged polymer, PAH, or polyamine hydrochloride, on a negatively charged organic monolayer. So we had positively charged dots on the background of a negatively charged monolayer. And because as I said, our sensors are very charge sensitive, you can see a very good contrast in the photocurrent image on the right-hand side. If we then expose that to DNA, what happens is that the DNA is of course negatively charged. It will stick to the positively charged PAH, and you can see that the photocurrent contrast all but disappears, because now you have negatively charged DNA on a contrast of a negatively charged surface. So theoretically, we could use this technique to interrogate DNA arrays without the need for labeling any of the DNA. So I could save some time, but the problem is there's a lot of established techniques out there, and it's difficult to break into the market with that sort of technology. On the left-hand side, you can see the work of Bo Zhou, who's also here in the audience. So he developed a calcium sensor. He used a different semiconductor, which I will come to back to later, hematite nanorods, and he put a calcium-sensitive layer on top of that. So that is PVC that contained a chelator that is sensitive to calcium ions. It's a crown ether. And when you look at the results here, you can see that the sensor actually responds very well to calcium, to changes in the calcium concentration, but it does not respond to changes of any other interfering ions. So these were two examples of the sensor work that we did. But what about living cells? Because I promised you living cells, so that's what I'm going to talk about now. So the first successful experiment, or maybe the first experiment that we were able to publish, was done by Devin Jan, and he used yeast. So yeast doesn't stick on surfaces, so to immobilize the yeast on the surface, we mixed it with a low temperature gelling agarose gel and mobilized the yeast with that. So you can see that we used a model layer that did not have any functional groups on it, so totally insensitive to pH or anything else that is floating around in the solution. So we should only get a signal from the yeast. And you can see in the optical image that we have a dot here that contains the agarose gel mixed with the yeast. And on the left-hand side, we have a dot that does not contain any yeast. And if you measure the photocurrent image of that, you can see the blob with the yeast, but you can't see the blob that doesn't contain any yeast. So that means the signal definitely comes from the yeast. And we can basically see, if we look at the photocurrent curves, we can do both of these things that I described to you before. You can see a shift of the curve to the right. This is because yeast cells, like most living cells, are negatively charged. So you can see the shift of the curve because of that. And you can also see a decrease in the maximum photocurrent because of the impedance of the yeast cells. Now, this is not single cell imaging. You can see the scale on there. This is 200 micrometers across one of these dots, so that you can actually see that with your naked eye. But I will now show you the work of Rachel, who's also in the audience. And basically what we realized from the experiments with the yeast is we needed a really good contact of the cells with the surface. So Rachel developed a setup that allowed us to apply a very well-defined force to press cells onto the surface of our sensor. In collaboration with Thomas Iskrach's group, we grew these organoids consisting of living cardiomyocytes, so cardiac cells that were actually beating cells, and we pressed them onto the surface. And if you take a photocurrent image of that, you can see the cells that are directly in contact with the surface by a minimum in the photocurrent. So you can see the signal, the photocurrent signal with time that we measured with these types of measurements. You can see the signals, the action potentials of two neighboring cells measured simultaneously. So one curve red and the other one black. You can see that these two cells are beating out of sync. You can see where one has the troughs, the other one has the peaks. And we then added a drug, Rachel added a drug, which is verapamil, which is which is used in some arrhythmias and also slows down the beating of the cells. You can see that now the cells are beating more slowly after the drug was added. And what you can also see is that these two cells are actually beating more in sync. And that's a really cool thing because nobody's been able to ever do this before, as far as we know, to measure action potentials of two cells simultaneously. So that theoretically we could measure, of course, the action potential of even more cells simultaneously if we scanned a larger area. So we tested various drugs, but I'm only showing you one example here. So when you look, we also did some work that was actually statistically relevant. So we can see that the verapamil caused statistically significant change in the beat period, and we could also see that the amplitude showed a significant change after the addition of that verapamil. So we tested other drugs and they also worked in the same manner. So that was a very success. We were very happy and proud of that work and was published relatively recently. But a lot of people are interested in looking at cells when they are cultured on the surface, or on a chip maybe. And the problem is when you culture a cell on the surface, the membrane, the negatively charged cell membrane, is not actually in touch with that surface. So generally, I apologize for the relatively simplistic drawing, but generally you have proteins sticking out of the cell membrane which form these focal adhesions, and what happens is that then there is a gap between the cell membrane and the surface of your chip, which is filled with electrolyte. Now, I told you our chips are very sensitive to charge, but the problem is, of course, the field that's caused by these charges decays within a so-called debye length. And the debye length in physiological salt solution is less than a nanometer. but these gaps between the surface and the sensor are tens to 100 micrometers. So you can imagine we cultured a lot of cells and we saw absolutely nothing. So basically the charges are screened by the electrolyte, making labs insensitive to these cell surface charges. So how did we get around that? Because I wouldn't tell you that if we hadn't got anything better to show you. So essentially what we've done in recent years, a lot of work has gone into that, is to measure, do photocurrent imaging with semiconductor substrates without an insulator. So we're not measuring capacitive currents anymore, but what we're doing is we actually measure Faraday currents, so proper oxidation or reduction currents. The first semiconductor we used was ITO, so indium tin oxide coated glass. We also played a bit with carbon dots and with zinc oxide nanorods, and we also used hematite. So I'm only going to talk about the ITO and the hematite today. So the ITO was almost an accidental discovery because we didn't really expect significant photocurrents from ITO. But essentially what you do is if you do a linear sweep voltammetry with the indium tin oxide that we had lying around in the lab at the time, you can see that when you chop the light, you get a nice photocurrent as you increase the voltage. So what's happening? Well, ITO is an N-type semiconductor. So when you apply a voltage to get a depletion layer on the surface of that semiconductor, you again promote electrons from the valence band to the conduction band, and this time the minority charge carriers that accumulate at the interface are the positively charged holes. And these can then oxidize species in the solution. And what we essentially did, because we worked in a physiological salt solution, a PBS buffer, we oxidized hydroxide. So with ITO, we concluded that we're just oxidizing it to the point of a hydroxyl radical, not exactly going to oxygen, but we basically oxidized the hydroxide. And if you look at it, because we're oxidizing hydroxide, these structures are very strongly pH sensitive. So we get greater than Lorentzian sensitivity for pH. And we also did some model measurements to carry out, to find out what the resolution is. So we estimated a resolution of 2.3 micrometers. I think if the experiment had been done slightly differently, we might have probably achieved the same resolution as with silicon. But the resolution was still good enough to actually work with living cells. Living cells are generally tens of micrometers across. So this is the first successful work where we cultured MG63 osteoblast-like cells on there. This work was done by Fan Wu. So she had tried to culture cells on silicon, didn't see anything, and then towards the end of the PhD, we came up with this ITO photocurrent measurements, and she cultured the cells on the surface. Here's the optical image in the middle, and on the left-hand side, you can see the photocurrent image. So you can see, we can see a clear photocurrent contrast where the cells are. So if you look a bit more closely, you can see that the photocurrent is actually smaller under the cells. So why is that? Well, as I already said, cells are negatively charged. So that will push hydroxide ions out of the gap because obviously you have electrostatic repulsion. And when you then turn on the light, apply a voltage, you will start to oxidize the hydroxide. The hydroxyl radicals will diffuse away from the surface as they accumulate at the surface, but now you've basically depleted the hydroxide on the surface, which means the hydroxide has to diffuse back to the surface. So you build up a concentration gradient, essentially. So, of course, the diffusion of the hydroxide into the gap is hindered by the presence of the cell, because you only have a narrow gap that it has to diffuse in. So essentially, the photocurrent under a cell is smaller because of the charge of the cell and because the hydroxide has to diffuse into a very narrow gap. So that is what gives you the photocurrent contrast. But the really good thing is now we can actually sense anything that is within the concentration gradient of our electrode. So anything that you build up a concentration gradient by oxidizing hydroxide, anything that's in that concentration gradient will have an effect on the photocurrent. So Fan also tried different cell types that were known to have different cell charges, and she basically correlated the photocurrent contrast that we were measuring very well with the zeta potential of the cells. So we showed that we can really measure surface charges of cells with this technology. But we sort of realized that ITO is really not an ideal semiconductor substrate, and Bo was talking to people in other groups, basically found out there's lots of people working on photocatalytic water splitting at the time. There were several groups in our school, and one of the materials that many people worked on was hematite. So he optimized the electrodeposition of a hematite film, and it gave a very nice photocurrent. So much better semiconductor. You can see we get a good photocurrent. The photocurrent is much larger than the photocurrent we measured on the ITO. The photocurrent was also much more stable. And you can see when you look at photocurrent measured with modulation at different frequencies, that we could also measure at relatively high speeds. So with the ITO, we could maybe modulate at a maximum of 10 Hertz. With this, you could modulate the light at the kilohertz. So that was for much faster measurements. And he then proceeded to grow cells on the surface. So this is another example where we managed to measure something worthwhile. So this is a measurement with P50 neuronal cells that we cultured on the surface of these hematite sensors. And you can see the optical image on the left and the photocurrent image on the right. And what Bo did is he added a surfactant. So what the surfactant does, it permeabilizes the membrane. So now the hydroxide ions can go through the membrane, and therefore the photocurrent contrast is all but disappearing from our photocurrent image. And this didn't actually kill the cells, so we kept the concentration low enough to not kill them. And when we replaced it with fresh buffer, removing the surfactant, you can see the photocurrent contrast is coming back, at least for some of the cells. Some of the cells died, other cells survived, and the photocurrent contrast recovered. So if you look on the left-hand side, this shows the whole experiment took 102 minutes, so it's a longer time. This was not in real time. But you can see that when the surfactant was added, the photocurrent increased, and then when the surfactant was removed, the cells recovered, the photocurrent decreased under the cells, while some of the cells did not recover. So photocurrent stayed high because the cells had died. So taking one of these images takes about 36 seconds. So that was the optimum speed that we determined at the time. And this was now not done with the modulated light, but this was actually DC scanning. So we had our mirror scanning set up. We continuously scanned the laser beam across the surface and recorded the photocurrent while we were doing that. And the optimum speed that you could use with hematite was 36 seconds. So good for some imaging, but maybe not fast enough for others. And that's where I wanted to show you some unpublished results that one of my current PhD students, Ruishang, has produced. So I thought this was an inherent problem with the hematite, but I was wrong. So basically, Ruishang optimized the fabrication of the hematite. I won't give you the details because it's not been published yet. But now we can do much faster imaging speeds. So if you look at this, basically on the left, this isn't done in real time. So on the left you see the movement of the laser beam. On the right you see the build-up of the photocurrent image. So that whole photocurrent image, 100 by 100 points, took 3.6 seconds. So it's, that I think is really quite good. So that would actually allow us, if you wanted to do, so for example, continuous line scans, we could actually measure things like action potentials again with the hematite. And hematite is of course a much cheaper substrate than silicon on sapphire. So it brings it into the realm of something that people might want to might want to use and buy. So. I also wanted to show you another of our latest projects that we haven't published yet, but I really think this is quite exciting work, so I wanted to show you at least some of that. Because a lot of times when I present at conferences, people automatically ask, well, you do this 2D imaging, can you also do it in 3D? And this is what Jarge is working on, one of my other PhD students, and she's basically making porous semiconductor structures, and the way we can get depth resolution with our photocurrent measurements is again to use a 2-photon excitation. So you understand why I was talking about that in a little bit more detail earlier. Of course, because it's hematite, we have a different band gap, we have a different wavelength, but we can actually get depth resolution in the hematite with that sort of technology. So what do we want to do with that? Well, we could use that to investigate variation in photocatalytic activity in mesoporous electrostructures, but we could also use it for cell signaling processes in 3D tissue culture, like organ-on-a-chip devices. So this is how Jarge is currently making these electrodes. So she's making she's basically self-assembling a template of polystyrene beads, 30 micrometers in diameter. Then she infiltrates it with an ITO precursor, fires it to get rid of the template, and then electrodeposits hematite. And then we have basically our semiconducting porous structure. And if you look at the picture on the top right-hand corner, which is the SEM image, we have nicely interconnected pores, so it's, the fabrication works very well. And here you have some very early experiment. We have some very early results where you basically have an XY scan, an XY photo current image, and then on the right-hand side you have an XZ photocurrent image of the same position. So you can see the pore on the right-hand side, there's a bit of distortion there that we're still working on, but you can see the pore shape in the Z direction as well. So basically the 3D imaging is actually working, so hopefully we can also publish that work very soon. So what does the future hold for this imaging technology? Well, the LABS micro endoscope for in vivo imaging would be something that I would like to develop. So this is where we've already done some preliminary experiments. Could be used for detection of cancer, or could be used inside the heart for looking at fibrillation when people suffer from arrhythmia. But we could also use it for inline sensing and organ-on-chip technologies. There is a big drive towards making organ-on-chip devices that can be used for the discovery of drugs and also for understanding physiological processes better. And we think that the photoelectrochemical imaging would be an ideal technique where we could measure cell responses in situ and in real time. Another possible area of application is to use this type of photoelectrochemistry as actuators. So there's a paper by a German group that was recently published that I was also briefly involved in, where they developed an actuator to control the pH inside the microfluidic channel. Because of course you oxidize hydroxide. If you deliberately have relatively large photocurrents, you can actually acidify the solution. And there are now, semiconductors, has also been published, that can be both N-type and P-type, so you can both increase and decrease the pH in a microfluidic channel. So it could be quite a valuable application for this. And you could also, of course, use it for the characterization of electrode materials for green energy applications. And that brings me to the acknowledgements. I would like, of course, to thank all the funding bodies that have given us money, because without them I wouldn't be able to do this work. I would really sincerely would like to thank all my collaborators over the years, because without whom the work would have been much more narrow and not nearly as interesting as what we have achieved to date. And of course, I would also like to thank the people who did the work and are still doing the work. So this is in chronological order. So this is a while ago, this is a few years ago, this is after COVID, this is very recently. I seem to be shrinking. That's how it goes. And I also would like to very much thank my family for allowing me to keep a good balance between my work life and my family life. And I thank you very much for coming and listening to me for all this time. And I'm very happy to answer any questions you have.
Well, thank you very much, Steffi. I think it will be clear to everyone your excitement and enthusiasm with electrochemical sensing and photoelectrochemical imaging. Thank you very much for that inaugural lecture. We don't have much time, but I could see if there are some urgent or pressing questions from the audience. I think the audience is saying that they would like a refreshment and they would like an opportunity to ask you questions over a refreshment. So Steffi, thank you very much indeed for a wonderful inaugural lecture. Thank you.